From Microbe TV, this is Office Hours. I'm Vincent Racaniello, and welcome to the small corner of the internet where you can hear about viruses and meet people that you've met here before. Nice little community. Welcome, everybody. And Office Hours is a kind of expansion of the office hours I do for my virology course every week. You get to hear it right from me here on the internet. So if you're taking the course, now's the time for your questions. But you can ask anything you want. I'll do my best. I've been working on viruses for 40 years, but I don't know everything. I'm going to have some, answers to your questions. I always get that because it's the same in my class. I get questions. Well, that's a great question. I love great questions. I don't have an answer to that. There are two things. I either don't have an answer or I don't know. Anyway, let's welcome our moderators tonight. I saw Steph, I saw Les, and I saw PDK. I think I got everyone. Oh, there's Frank. Hello, Frank. Welcome. And thank you for moderating. I have to say at the outset, I had a pretty miserable day today, but I'm already cheered up seeing all of you here tonight. So I had immune recording fail. Fortunately, I had a backup. My lecture recording failed. No backup. Have to repeat it here in the basement one night in the next couple of days. And then tonight the train was late, making me anxious, but I'm here in time. So it's all good. My spirits are raised by your presence. Let's see. Did I miss anything? I think there's an important thing. So I went to see the theater yesterday. Is it yesterday? Yeah, I think so. For TWiV 1000, it looks great. I said I want it, so she gave me a price. I have to fill out a form, and it's hours. So if you're interested in going to TWiV 1000, it is the theater is lovely. Helen Mills Theater. It's a couple of blocks from the incubator. Very convenient. It has a seating for 130. And so I don't think we're going to fill the room. So if you want to go, just send an email to vincent@microbe.tv, put TWiV 1000 in the subject line. And we are asking a donation of $100 per seat. You know, it's like the Metropolitan Museum. We suggest you give $100 per seat because it's pretty expensive to book a theater, you know. I don't know if you've ever done that, but yeah, especially theaters over 99 where the prices go up. But yeah, if you want to come, Vincent@microbe.tv. All the TWiV crew will be there. That's me, Dixon, Alan, Rich, Kathy, Brianne, Angela, and of course Daniel. All 8. And we're going to come up with an amusing program for you, which is now our next task. So if you want to go, Vincent@microbe.tv. Should be fun. And that's the last big celebration for TWiV numbers. April 15th, 2023. It's a Saturday. It'll be at 6 p.m. Okay. And after that, well, you know, it took how many years? 13, 14 years to reach 1,000. I don't think I'm going to be around for 2,000 because if we do 152 weeks a year, 100 a year, that would take 10 years to get to. Yeah, I'd be around for 2,000. Yes, sure, absolutely. You know, Raquel Welch just died today. She's 82, so I'll be around for 2,000. It won't be streamed, no. We want to make it, you know, a unique event for participants. But it will, of course, be recorded. Oh, and Dixon's nephew is going to bring his trio, and they will play before and after the event. And then we'll have you to the incubator afterwards for a little reception. So if you want to meet everyone, there you go. April 15th. Vincent at microbe.tv for more information. So I guess you guys like my my picture in front of my license plate, right? Get some comments there. I've had that plate for quite a while now. And did it get looks? Yeah, it gets looks all the time. But in the last year, 2 years, people have been photographing it. I pull up to a light and I see people taking pictures. I have never seen any of them. Nobody posts them on the internet. So that's good. I like that people take a picture and they think of virus for a fleeting moment. There you go. So let's see. I told you who's here from. Let's see where people are from. Some of you put in where you're from. So Lise is from Columbus, Ohio. I will go through quickly just to see where people are from. Nicola is in Italy. And Nicola, my first graduate student, was Nicola from Italy. Oh, that was 1984, I guess, something like that. Wow. A long time ago, many, many years ago. Let's see, who else is here? Claire is from the UK. I know that our moderators, Steph and Les, are from California, and Frank is from New York area, and PDK, I don't know where you're from. No idea. Brian is from Montreal. Simon is from San Jose Bay Area. Lisa is from Oswego, New York. Just a few. Just a few. I'm just curious always to see where people are from. Then I will scroll back and start hitting the questions. And look at this. We already got a little contribution from Lisa. Thank you very much for your contribution to science education. Khan. Wow, interesting name. Sitting outside in Orlando, or I guess it's warm. Charleston, South Carolina, the home of Michael Schmidt. How about that? Boulder, Colorado. PDK. I don't know where you're from. Yeah, but it's a bad day, but I'm good now. It's all good. South Jersey. All right, when is TWiV 1000? April 15th. April 15th. Okay, tax day. Just remember that. Tax day. Eastern Massachusetts. Eastern Massachusetts. Yeah, Amy's going to be at TWiV 1000 for sure. Absolutely. Santa Barbara, Colorado. Silvio Pina, thank you so much for your contribution. Santa Fe, hello, Noir. Welcome, Santa Fe, New Mexico. Northeastern Ohio. Westchester, New York. Lake Tahoe, UK. Tucson. Sechelt, is that how you say it? Houston, Texas. Michigan, with microbe TV stickers taped inside your car windows. I hope you don't get a ticket because of my sticker. Vashon Island, Washington. That's cool. Westchester, hello, Elena. Los Angeles, Hamilton, Ontario, Central Florida, Anaheim, California, Newcastle, Buda. Hello, Neva. Some of you folks, I'll be in your neighborhood this summer. Take a look at the schedule. There's a calendar at microbe.tv/calendar. And if I'm around, we could meet up. Ontario, Kentucky, Frederick, Maryland, Alberta, Toronto. Ooh, Ukraine. How's things over there? Everything good? Are you all right? Thank you for joining us. I appreciate it. I think you're the only person from Ukraine here tonight. Ontario, Toronto, Saratoga, New York, Albany, West Virginia, Northern Virginia, Atlanta. I'll be in Atlanta twice. I'll be first in April, I believe, for a clinical microbiology conference. Yeah. No, I don't want to do the updates tonight. Thank you very much. Where did we go? Atlanta, LA. Thank you, Lisa. Oh, you did another one. Thank you very much. I appreciate it. Cairo, New York. Shoreview, Minnesota. Boston. Turkey, hello. Oh, good. I'm glad you're safe. Bangkok. Oh, my gosh. Holy cow. Watching from Long Island. I just think it's awesome. Canada, Berkeley, and Silver Spring, Maryland, not too far from Amy. Hello, Amy. She must be listening. Yeah, the family is worldwide, isn't it? Isn't it great? Loving it. Hello, Vanity. Welcome and thanks for modding tonight. Do you have your feet on the ground? All right, let's go back to the beginning and see what's up in the virus world. Let's see what we have here. Excellent discussion with Scott Hensley. Yes, he was good. And I want to line up more discussions because I think we can do the virus discussions better than anyone else. By the way, we had on Immune today a really good discussion with Jonathan Kagan, I don't remember his name. Damn. He was really good. Unfortunately, the recording is not going to be as good as usual because it's screwed up. But glad you liked it anyway. Okay, Polio Pete. Welcome, Polio Pete. How reliable is the Israeli study that states Paxlovid showed no measurable benefit in adults 40 to 65? They were looking at hospitalization, I believe. So here's the problem. It's an observational study where they pull hospital records and they say, okay, this person got Paxlovid and this person did it. What happened to them? It's not a controlled study, which is what you'd like. Why is that an issue? Well, there can be other confounding factors that you haven't controlled for. When you enroll in a randomized controlled clinical trial, an RCT, the gold standard, as Daniel says, for figuring out what something does, you take a control group and a treatment group and you make sure that other stuff, as far as you know, doesn't play into it. Like none of them have cancer that could affect your outcome, right? Stuff like that. And you don't know that in this kind of a study. So I'm skeptical of the outcome because many other studies have shown that there is a considerable benefit to giving Paxlovid early on, especially to older people with comorbidities. So that's the story there. That's what I think. And I have not seen the actual data, okay, Polio Pete. So what is it that we say in the business? It's my opinion without seeing the data, but knowing the limitations and knowing what other studies have found. But we'll certainly take a look at it. Probably Daniel will. So grain of salt, reliable, right? Okay, let's see. Last week, Dixon said blood supply isn't tested for Chagas, but I'm rereading The Kissing Bug, and in it the author says, yes, the blood supply is tested at least one time. So let's look it up. Is blood tested for Chagas? You know, Dixon is the man. No, the blood supply. See, that wasn't the right thing to do because blood is tested if you're suspected of being sick, right? It makes sense. Donors are tested once, and if the result of their test is negative, they will not be tested when they donate again. There you go. So, Patricia, the book is right, and Dixon is not. But it happens. Should I tell him? It's not the first thing he's gotten wrong. I've gotten things wrong as well, so we won't worry about it. Thank you for clearing that up. Excellent. How many different animal species have been known to get infected by SARS-CoV-2? Oh my goodness, tons. And we're doing a paper tomorrow on TWiV that lists many of them. So let me pull it up here because it's in the TWiV papers folder. And what is tomorrow? TWiV 985. All right, so this is an interesting paper about extent of infection in deer in New York State. And let's see if they talk about other animals. Yeah, they don't. Many dozens, I think, so far have been shown to be infected. Wouldn't you say, Amy? Dozens of mammals have been shown to be infected. Deer are extensively infected. The paper we're going to do tomorrow shows that many of the older variants, like alpha and beta and delta, are still in the deer in New York State, just circulating among the deer. I guess none of them take off or get out competed. I don't know what that means, but it's interesting that they're there. And many other animals are infected as well, right? We got minks and ferrets and many, many more. So the exact number, I don't know. There are papers that we've done where they list them all, but I don't have it at the tip of my tongue. If someone does, maybe Amy's looking it up. I got her here on the text, so she could always weigh in. Okay, that was Elena. How safe is it for the kidneys to keep getting Remdesivir after 1, 2, 3 infections? You know, that's a question for Daniel. Let's see. When am I doing Daniel's? Daniel's tomorrow. It's Thursday. So if the sticky yellow people can you write it on the sticky and send me with the summary? Ask Daniel, is it okay for kidneys to get multiple doses of Remdesivir? I guess if you have issues, comorbidities and so forth, you want to get it every time you test positive, right? Wow, lots of good questions already. Just finished the Cutter incident. Thanks for suggesting it. Cutter incident is great. Here it is. Cutter incident. Paul Offit. Really good story of, I know I showed this last time. The story of the initial rollout of inactivated polio vaccine in the 50s. And the initial batches were incompletely inactivated. So that's the story of what happened. So read it. It's really good. I like it. Okay, Lori says, when B cells present antigen to CD4 cells in the germinal centers, do they do so via antibodies? So the B cells have antibodies on their cell surface. The antigen binds the antibody, and it can be taken up, processed, and then presented to CD4 cells. And they also the uptake, the interaction with the antibody on the surface of the B cell will also cause cytokine release. I'm gonna pull up a slide from my class just to make sure we're getting this right. So roughly, what lecture would that be? Would that be mechanisms of pathogenesis? No. Acute infections, persistent infections, transformation, vaccines? No, we have to go earlier. Adaptive immunity. Here we go. This will have the picture you want. Since this is office hours, we do have the opportunity to, here we go. Here's the exact image I want. Now, let's see if I can get my screen share thingy up here. Here we go. There you go. Look at that. Oh, I spun through it. So I'm going to make it a little larger because it's kind of small. All right. So there we have, it's still kind of small, right? Let's see if we can fix that. We have to hit the Option key to get rid of that, and then we can move it. All right. So here we have a B cell, and we have antibodies on the surface, right? And so those antibodies, specific for an epitope, one epitope, they are binding the antigen. And the antigen, you can see, is taken up, processed, presented on MHC2 to CD4 cells. And then the CD4 cells are going to make helper cytokines that help the B cell differentiate into a plasma cell that then makes a lot of antibody. All right, so now I have lost your question. Yeah, it's via antibodies. There you go. Okie dokie. Okay, Kang. Hello, Kang. Mentioned in previous TWiVs, the FC region of antibodies binds to effector cells, can ameliorate disease, but does the Fab still need to bind viral epitopes? Yes, and in fact, we have some, I believe we have some pictures of that here in this same lecture, so let's pull it up because it is timely. And so just so everyone knows, this is truly office hours. I'm showing slides. Look at this, screen share. There's an antibody, right? You have the Fab or the antigen binding region, and then the FC region, which binds FC receptors and can do other things. So what was the slide that I was looking for that shows, like antibody-dependent cyto... Oh, here we go. Antibody-dependent cellular cytotoxicity. So this is one kind of FC-mediated effect. So we have antibodies binding a virus-infected cell. So you see in this case the Fab is engaging antigen on the cell surface. Then the NK cell has receptors for the FC portion, FC receptors. And when this is engaged, the NK cell kills or induces apoptosis, programmed cell death in the infected cell. So that's how it works. Yeah, you do need to have antigen engagement. Yeah. Good. These are good virology questions. I like this. Things I can pull slides out. That's great. What's the difference between MERS, SARS, and SARS-CoV-2? All right, let's start with SARS. They're all coronaviruses. They are epidemic human coronaviruses. They've caused outbreaks, right? And before 2003, all the human coronas caused common colds, mild infections. And then 2003, suddenly an outbreak of severe acute respiratory syndrome, SARS. So they named it SARS coronavirus. And that was limited to about 8,000 cases globally because there was not a lot of asymptomatic shedding of virus. So people weren't walking around shedding virus because half of the people got really sick and they were in the hospital. And when you had peak symptoms, that was peak shedding. So there was no pre-symptomatic or asymptomatic shedding. So easy to confine. Then 2012, and by the way, SARS came from a bat which infected probably a civet cat in the fields somewhere. The civet cats were brought into the meat market and they infected people. Then MERS, 2012, an older gentleman in Saudi Arabia or Egypt, I can't remember where, Saudi Arabia, sorry, dies of severe pneumonia, and it turns out he's infected with another coronavirus, not SARS, but related, and beta coronavirus. And so this is called Middle East Respiratory Syndrome coronavirus. So very similar disease, severe lower tract infection, and that one did not do very effective human-to-human transmission. It remains to this day a sporadic infection, a few a month, mainly in the Arabian Peninsula because it comes from camels. So that virus probably jumped into camels from bats many years ago. And camels largely are all infected at birth or shortly thereafter. They get a mild respiratory infection. And camels are very popular in many parts of the world, particularly the Arabian Peninsula. So people can get infected. They tend to be older people, but not all. And not everyone has a camel exposure. So how the others are infected is not clear. But they're not long chains of human-to-human transmission. So that one continues. There have been many thousands of cases and, at least a couple every month. And in SARS-CoV-2, and these viruses are all sufficiently different to cause slightly different diseases, and they have genome sequences that distinguish them. So SARS-CoV-2 related to both, but not the same virus. Can I pull up a phylogenetic tree of these viruses very quickly? Where would I have that? I don't know. I'd like to show you a phylogenetic tree of the human coronaviruses. Let's see. Phylogenetic. This may be dangerous because I might get garbage doing a Google search. Yeah, I remember this one. Here's the one from. It's not going to totally answer your question, but it will get partial answer. So here we go. Let's do another screen share. So this is from this paper, Characteristics of SARS-CoV-2, October 2020, from Zhongli Shi's laboratory. So this is, a comparison of the genome sequences. And here we have the original SARS here, right? So these are all beta coronaviruses, and that's your SARS, original SARS-1. And then in a separate group, we have SARS-CoV-2 here, some early isolates. So you see they're on a separate branch altogether than SARS, so they're different enough to do that. 80% genome identity. And here's MERS down here, which is a Merbeco virus. So there's subgenera here, Sarbaco. So the SARS and SARS-CoV-2 are Sarbaco, SARS beta coronaviruses, and this MERS beta coronavirus is down here, so on a separate tree. So that's the relationship between them. Good question. All right. And someone has said, there was a question way at the bottom. Where is it? Ooh, so the antibodies that present to CD4 would be part of the group of non-neutralizing. No, not necessarily. They could be either one, right? It could be anybody. All they have to do is bind virus. They don't have to do anything beyond that, because at that point the antigen is just binding the antibody on the surface of the B cell, right? New York Times today has an article comparing different estimates of COVID deaths since ending the zero COVID policy. Okay, I didn't see it. Let's, I don't want to look it up now. What does it say? Someone summarize it. Read about the Marburg virus outbreak in Africa. Yes, and there's not a lot, but we're going to discuss it briefly tomorrow on TWiV. So we're recording TWiV tomorrow, which is an unusual day. And let me see if I can bring up, when I looked at it the other day, there was just a WHO report. And there may be more since then. In fact, I'm sure there's more. But let's see. Okay, what's the story here? $9.85, TWiV 985. There you go. And the article, Equatorial Guinea confirms first ever Marburg virus disease outbreak. Let's share this with you. Let's get it so you can see it. There you go. Equatorial Guinea, first ever outbreak. So Equatorial Guinea is on the... All right, I'm going to say the wrong thing. Yeah, it's on the east, the western coast of Africa. Okay, and first outbreak. How many? 9 deaths and 16 suspected cases. So, I'm a little surprised that it's there because I didn't know that. We know the bats that harbor Marburg virus. In contrast to Ebola, actually have found infectious Marburg virus in a specific kind of bat. Let's find what kind of bat. Host of Marburg. So none of this I carry in my head, but I know where to look for it. The host is the African fruit bat, Rosetta's African fruit bat, Rosetta's aegyptiacus. So, what is the range of Rosetta's aegyptiacus? Let's look that up. Range. See where it is in Africa. Do you think we could get a nice image of the range? Yeah, we can. Look at this. Wow. I'm going to click in. They're not going to let me see it because they put it behind a paywall. So here's the range of this bat. Where's my screen share? And so on the left is Rosetta's aegyptiacus. Here you go, and Guinea is. Which of these is Guinea? Let's take a look. All right, now I need to look at another. Equatorial Guinea is north of Gabon. It doesn't look like the range is Equatorial Guinea here, right? If I'm interpreting this, it's the square one, right? Doesn't look like it should be there. So that's why I'm kind of surprised that the outbreak is there. But it may very well be that it's in a different kind of bat. I don't know. So I don't think this is going to go very far. I think they can contain it, but it is surprising to me. It is surprising. Yep. Hello. Is the Dr. Francis you talked about on TWiV the same Francis? Yeah. Thomas Francis ran the 54 polio vaccine trials. Yes, absolutely. And he's certainly in the Cutter incident, right? Let's take a look and see how many Francises there are. Field trials, Francis Thomas, field trial report. Yeah, definitely. He was shall we say, a giant in virology. He's a very smart guy, very outspoken, kind of a bully. I didn't know him, but this is what I've heard. But yeah, that's the same person. He really did a lot. He was really a founding person in virology. I don't want to say father. I don't like to say founding father. Founding person. How's that in the field of virology, to conceptualize it and put it in scientific terms? It's a lot of good stuff. Elena, what was your earliest big breakthrough discovery in your virology research? Well, big breakthrough? Big breakthrough? All right, so I did some cool stuff with flu, but let's make it big. I've been lucky to have a couple, right? So with David Baltimore, I showed that a DNA copy of poliovirus RNA was infectious when inserted into cells. That was big because everybody now makes infectious DNAs of their viruses. That was the first one. Nobody knew it would work. So that was cool. And then in my lab, second big breakthrough, we identified poliovirus receptor with Kathy Mendelson, a student in my lab. And third big breakthrough, we made transgenic mice with the human poliovirus receptor gene, and they are a new model for polio. And that was with Rebow Ren. And the three, what I would say, are big breakthroughs. And then the fourth is Microbe TV, TWiV, and all the podcasts, which I think are cool. Not breakthrough because I didn't invent anything, but I just thought it would be cool to do science podcasts at a level that nobody else was doing. People were doing science podcasts before me, sure, absolutely. But I wanted to have scientists talk in scientist terms, have conversations, and let people hear them. Nobody thought of that before. That's not a big breakthrough, I agree. But I really like it because it's what I'm doing now, right? What do you think of the Cochrane review that says wearing masks in the community probably makes little or no difference to the outcome of influenza or COVID compared to not wearing masks? I think this is a difficult conclusion for a number of reasons. First of all, it's a meta-analysis, right? And, you lump together a lot of bad studies with good ones, and the outcome is a bad meta-analysis. So I would prefer to look at the individual studies. And I think it's really hard to do those studies, right? We've seen a few where the outcomes are marginal because it's just difficult. It's difficult to get people to wear them properly. But there are plenty of studies, which, for one, I remember that was very impressive, where they went into schools and when they compared rates before and after masking. So when kids were masking versus taking them off. Now, admittedly, it's different times of the year. I totally agree that hard to compare, but it's the best you can do. And that had an impact. So I just am suspicious of some studies that would say masks don't make any difference. It doesn't make any sense, right? So there has to be something wrong with that. Peak says, my parents said I'm already going to New York City in the summer, so I can't come. Why not? You're going to be in New York City. Why can't you mean you're going to go in the summer, and April is not the summer? Where are you from? You Montana or something? It's too far away? I would never miss Wednesday if I could avoid it. Yeah, I enjoy it. And by the way, folks, next week is the 22nd. You know what that is? It's Q&A returns for its monthly appearance. So those of you who need your Amy view, there you go. You're going to get it. She'll be back next Wednesday. It'll be fun. The seat donation, if you can't do it and you want to come, we're not going to kick you out. But you do need to get your name on the list because there are only 130 spots, right? Will I be at the incubator on Monday? Well, I think so. I have class on Monday. My plan is President's Day, but we have class on President's Day. It is not a class holiday at Columbia, so I'll be there. But you should just check Sunday sometime. Just shoot me an email, okay, to make sure I'll be there. There's just something going on that might prevent, but I think there's a 90% chance I'll be there. But just check my email Sunday. And I am working on getting some other guests for office hours as well to mix it up. Yep. And by the way, you may not be interested, but I worked with Ray Ortega for many years from ASM. We do the live TWiVs and TWIMs at ASM meetings. We're going to be in Houston this year. If there's anyone in Houston you want to meet up, that would be cool. We're going to do a TWiV and a TWIM in Houston. And anyway, Ray does the audio and the video. He's coming to TWiV 1000, and he's going to do the video. So that'll be fun. Is Amy's suggestion that this pandemic would last 5 years still on track? You have to ask Amy next week. What it's going to be it's the 2021 to all of 2020, 21, 22. That's 3 years. So we're into the fourth year. I don't know. What do you think, folks? Are we still in a pandemic? This is one of these non-virological things. So here we have, there's still a lot of deaths every day, right, from COVID. But I don't know if it's going to change, right, anytime soon. So in some countries, there's still more, right, and they're not in control. So in those countries, it's an issue. I mean, declaring a pandemic is all about trying to control, because the virus can be absent or low in some places and high in others. And how does that fall into the pandemic definition? But early on, certainly it's an emergency everywhere. But now I think not everywhere. And we're in a phase where we're trying to figure out how to move forward. So people are back to work, right? More or less normal lives. Most people not masking, over 90% immunity in the U.S. So no pandemic here as far as I'm concerned. So it's not 5 years. But in other parts of the world, it's continuing. And so it probably is going to be 5 years somewhere, right? Somewhere in the world, it's cocktail hour, as they say. Amy will be at TWiV 1000. Absolutely. She would not miss it. So if you come, you can meet Amy. I know a lot of you would like to do that. But it'll be April. She's not going to have any cashmere, I guess. Oh, maybe. From what I understand about the Cochrane mask study, which we just discussed, it was more a critique of the studies themselves than a conclusion masks made no difference. Okay. If you say Cochrane mask meta-analysis, let's see what the headline says. Physical interruptions. Yeah, here it is on the Cochrane website. Let's see here. Oh, yes, you're right, Patricia. The high risk of bias in the trials, variation in outcome measurement, and relatively low adherence with the interventions during the studies hampers drawing firm conclusions. That's right. There's uncertainty about the effects of face masks. So they're saying their uncertainty and probably in part, and maybe a major part, due to the problem with the design. As I mentioned, it's really hard to do those studies. It was a used car, actually. A couple of years ago, my Beamer had 250,000 miles on it. It was dying. Oh, my God. It was costing so much to keep it going. So I bought this used Mini. And now I don't drive to New York anymore. It's great. I drive like 3 miles a day. So I'm very, very happy. I wish I had done that with my Beamer, because the Beamer was a far better car. But the Mini's not bad. But it's a 6-speed manual, so it's cool. But I don't drive it. I don't drive 72 miles a day anymore. That's just insanity. I was insane for 38 years. It's too bad. If you go to the Ernst lecture, so I got the Richard Ernst Medal last year for science communication, you will see the presentation. Before my talk, a French horn quartet played a piece that was written for me called CD155, which is the name for the poliovirus receptor. And the first 3 notes, or 4 notes, or CD155, 5 notes, or CD155. I actually have a copy of the music. If anyone plays French horn and they want to record all 4 parts and mix them, that would be cool. I don't know if the saxophone guys can play that. It's a French horn quartet. Probably not. What do I know about music? Anyway, it's a cool piece. And that the fact that they did it for me, which was really nice, but that they based it on CD155, the poliovirus receptor, which was discovered or identified as the poliovirus receptor in my lab, that's also really cool. You got to leave the U.S. to get that kind of honor, right? No man is a prophet. No person is a prophet in their own land. That's absolutely correct. Not that I need to be honored, okay? But it would be nice now and then. And the Swiss got it. And they picked me before COVID, before any of our COVID programming. That blows me away. I think that's really cool. First opportunity to watch live in nearly 2 years. Now, I guess you've been working or you're busy. Well, welcome. And this isn't Q&A, of course. This is something new, but Q&A will be back next week. Thank you, Silvio Pina, for your contribution. I think I mentioned you before, but Dixon, I think it's a trio. As far as I've been talking to Dixon about it, and I think it's a trio. His nephew plays saxophone. I don't know what the other 2 would be. But they will play before and after the TWiV 1000. And they'll come back to the incubator and play there as well. Right? That would be cool. Nicola was a toddler in 84, so I can confirm he's a different. Yeah, you have the different last name, T. His would be L. Nicola LaMonica he's a scientist. He's published, so I can say his last name. Very cool. Would a Novavax series after the mRNA series confer broader, longer lasting protection for high risk? I don't know. I don't think there's any way you could predict that without doing a trial, and I don't think that's going to be done. If and you're talking about the mRNA series, you're talking about the 3, right? The 2 primes and the 3rd boost. I think if you wanted to boost, you could do Novavax. There wouldn't be harm in that. But whether it's going to confer longer protection, it's being looked at, I'm sure. And so we'll know the answers at some point, but I can't answer it. I don't think we could even predict. Oh, Elaine is going to be a TWiV 1K for your birthday. That's wonderful. Happy birthday. TWiV 1K, that's a cool way to put it. I like that. Here's an interesting town name in California, in L.A., Louisiana, sorry. The town of Our Lady of the Queen of the Angels of Porciuncula. Wow, that is an amazing name. Yes, Megan. Sorry, Megan. APHL is LabCon in Atlanta in March. Thank you. I can't believe you're here, so you got me to say it right. APHL is LabCon in Atlanta. That's where I'm going to be in Atlanta in March. I said April. I totally screwed it up, and fortunately, Megan, one of the co-organizers, is here and set me straight. So we're going to do a TWiV there. Kathy Spindler's joining us. So if you if you want to hang out, let me know. I'm laughing at another email. Oh boy. But I can't share it with you. Sorry. Bangladesh. I think we skipped Bangladesh before. Bangladesh, one of the last places where there's wild polio type 1. Are you and Daniel still making an infomercial about Paxlovid? Yes. Yes, we are. We're making part 1 tomorrow, actually. And I have to say, remember, I had a discussion with you guys, and I decided not to accept any compensation for that, and Daniel joined me in same, so that's really good. What do you make of Brett Weinstein's vehement anti-mRNA COVID vaccine crusade? I would say, and I'll be mild here because it's not the end of the show where I ranted last time. I'm calm. I don't know why I'm calm. I had a bogus day. First of all, there is no reason to rant against mRNA vaccines. There is no scientific reason. There are side effects, for sure. There's myocarditis, but it's quite rare, and it's lower than myocarditis that you get from COVID. There are no other known long-term side effects. There's no integration of DNA into your genome. So I don't know what he's talking about, but you have to put it in perspective. He's not a virologist. He is an evo-devo person, right? Developmental evolutionary biology. So he doesn't really know. He's not staying in his lane. And I don't think, and he's really embarrassing himself because he gets a lot of publicity. He's very highly exposed, and he says things that don't make any sense, that are just flat out wrong. And people think he's an expert, but he's not. So I could never do that. Say things that are flat wrong. It's like saying frogs have only one leg, right? And being on CNN and saying, yeah, it's well known that frogs only have one leg. That's a stupid example because everyone knows. They could look at frogs and know, but you get the point. Anyway, yes, it's embarrassing for him, and he's wrong, but a lot of people think he's right. So the mRNA vaccines are great. My whole family had it. Amy, everybody I know had mRNA vaccines. I had 3 shots. They're great. That doesn't mean that anything, an N of 5, but I have trust in them, and so do many people that I know. And Brett Reinstein is simply wrong. Okay, folks? And did our friend debunk him? I don't know if he did or not. He should. I think he did, actually. And he does a point-by-point job of saying why he's wrong. And in fact, John here says Brett debunks himself by having no virology knowledge. If you take the free course on this channel and then watch or listen to any Brett, that becomes clear. Thank you. Yeah, I mean, it just takes my virology course, folks. Now, I know it's long. It's 25 lectures. They're 75 minutes each. And you're not going to remember the first one once you get to the 5th one. But I still think you'd learn a lot and realize why he doesn't know what he's talking about. Do you think there might be any potential in investigating an mRNA-based vaccine for spike combined with other antigens such as N? Yeah, I think it would be worthwhile. I mean, people have explored other kinds of vaccines with N, but none of them have come to market. Because right now, it's hard for a new vaccine to come in. You have to show that you're better than current availability, current practice, as we say. So it's hard. I think it's worth putting N in, yeah. And I'm sure people are working on it for sure. I wish Amy were here. What defines a serotype? Okay, Amy, make sure I get this right. She's going to be waiting. She might be actually exercising now. The original definitions of serotype were done using poliovirus, protection against disease in humans or in animals. So they would say these people have been infected with this isolate and then they still got infected with another, or they didn't. Or in monkeys, you could infect and then challenge with another isolate. So they were based on animal models. Now, we move into the modern era. Many viruses there are no animal models for. So as Amy would point out, rhinoviruses were classified into serotypes. There weren't any animal models. Well, actually, the earliest studies of rhinovirus, as Amy pointed out to me earlier this week, were done in human volunteers, and that's how they started to get some of the serotype difference. But subsequently, it hasn't been done. So that's the classic definition of serotype, is that you have 3 viruses. If one gives you protection against only itself and not the other 2 in an animal model. And disease, you have to look for disease. But today, many people use neutralization of infectivity in cell culture, which isn't the original definition. How worried are you about H5N1? So here's the thing. I made a short. So on YouTube, you have shorts, right? You can take any video and you go to your phone and you hit short and you pick a part of it. My phone only limited me to 15 seconds. And he pauses and he says, yeah, I am. I thought that would be a powerful short to bring people to the original video. It got a lot of views. It got over 4,000 views in a short period of time. And then he says, I'll tell you something. This world is not ready for another pandemic. And that was it, 15 seconds. And people complain, well, why is he worried? And 15 seconds, what can you do in 15 seconds? But the point of a short is it grabs you and brings you to the original content. So I want to make more of them. Right? Every TWiV I could make, every pod we do, we could make shorts. And that could drive interest, don't you think? So, yes. Am I worried? I'm a bit worried because it's infecting mammals, right? It's infected humans before, less than 1,000 human infections, or roughly 1,000 with 600 deaths in humans. So it's infected humans who are mammals, right? But the mink and the sea otters, right? Or sea lions, I don't remember. I'm a little bit worried that it's acquiring mammalian characteristics. So I used to not be worried about H5 at all because it had been around for 50 years and never did anything. But this year we have so many bird infections. And you saw what happened when SARS-CoV-2 started to infect a lot of people. You generated variants because you have so much reproduction and so much mutation. So shorts are good, Kathleen, because it drives traffic. I need to drive traffic to my site, okay? You may not like it. You're not my target audience. I need people who have never seen TWiV before. They see this and go, oh, what is this? And then get hooked on the longer stuff. All right, I need to get traffic. I'm not going to survive as a science communication company without more traffic. It's great to have 117,000 subs. Not going to work in the long term. Okay. Yes, so I am worried about H5. However, Scott did say he's making a H5N1 mRNA vaccine. So we'll be ready, I think. It's good. It's good. I like it. Suggestion for TWiV, Dr. Jeff Ravitch, FC receptors. I know Jeff Ravitch. Yes, I do. Yes, that would be good for immune, I think. Yeah. Are all viruses contagious? I don't know what you mean. They have to be contagious. Otherwise, they're gone, right? They have to find a new host. So in nature, viruses have hosts. In our lab, we have cells in culture that we grow with, but that doesn't mean anything to a virus's existence. In nature, viruses need to go from host to host, from plant to plant, from mink to mink, or from frog to frog, whatever it be. They need to find a new host. Therefore, they're contagious. Now, are all viruses contagious? Yeah, they are. They don't all cause disease. Maybe that's what you're thinking about. But they're certainly contagious because that means to me finding a new host. Okay? You get your active immunity from being exposed to a deadly virus. How do you expose people to it without an animal? At some point, viruses become human viruses. We're animals, right? But you're talking about non-human animals. At some point, somewhere in any virus's history, any human virus's history, the virus came from an animal, infected people, spread it among people, and became a human virus. So animal to human spread is no longer required. And there is none. All human viruses began as a virus of some other animal. And it could have been a non-human primate. It could have been a bat. It could have been Homo neanderthalensis, right? It could have been a hominid. But they all start somewhere else. Late because I was roped into conversation with anti-vaxxers, anti-science. Second today, I need skills for talking to them. Calmness. Calmness is the key. You have to ask them, what are you worried about with vaccines? Why are you so opposed to them? Just calmly asking them, tell me what your concerns are, and try and address them one by one. And hopefully they'll be calm too, because they tend to get defensive when you come up with science and try and explain it that way. But you first have to be calm. It's really hard to stay calm because they get very testy. And they say you're a pharma shill and you're getting money from pharma and all this stuff. But use your science knowledge and say, what's the matter? Why don't you want to get this vaccine? What do you think is the problem? Well, it kills everyone. Okay, let's look at that statement, right? That kind of thing. Let's look at your statement. It kills everyone. It doesn't kill everyone because many millions of people have gotten this. You have to have numbers like that ready. But really calmly and folksy, you can use your personal experience sometimes to do it. That really helps. The more personal, the better, because you're another human that they can relate to. And I think those are, I mean, there are many, many professionals who have great suggestions like that. But that's what I would do. I would do that. What are the symptoms and the rate of mortality in deer? I think that the symptoms are respiratory infections, very mild, runny noses and that sort of thing. Let me see if we can pull up that paper again and see if they quickly talk about the symptoms. I don't think it's particularly virulent in terms of mortality. All right, here we go. Deer. Prevalence 30 to 40%. Viruses in respiratory secretions and tissues from several sampled animals. No, they won't really talk about it in this paper. My understanding is that they don't get very sick. But in a deer, if you kind of slow down for a few days, you could be vulnerable to predation, right? Let's see if they say it in the discussion. No. Nope. They don't talk about it at all. We'll talk about it on TWiV tomorrow. How does norovirus spread? Okay, so it's a fecally excreted virus. So it is spread like polio is, where you have poor hand hygiene. You don't wash your hands after going to the bathroom, and then you touch someone else and contaminate them, and then they put their hand on their mouth and they eat the virus and they get infected. You only need about 15 particles apparently to get infected. So the other vector of spread seems to be vomitus. There have been some outbreaks on airplanes where vomiting into the bag seems to be associated with multiple infections on the plane that are not all compatible with contact with say the bag, right? So if a flight attendant took the bag and contaminated his or her hands, that would be one way to spread it. But there's some evidence for because when you vomit, you're spewing out potentially virus particles, which can then, I hate to say this, you could inhale and swallow them. I mean, they're not going into your lung, but they're going into your gut tract. So that's how it's spread, but mainly by fecal oral contamination. Treatment. Right now, we don't have a vaccine, but we do have, you have to stay hydrated because our flight attendant Vanity says, great. Just one word, great. Yeah. So Vanity, if someone's throwing up in a bag, stay away, go do something else. Make some coffee. What was I saying? Treatment. So just keep hydrated because death will happen if you're dehydrated. That's the only treatment that we have. Yeah. Okay. Let's see. The last TWiV was super interesting. I've been planning on getting a flock of chicks. Would you or any expert hold off for a while given H5N1? Where are you going to put the chicks? Are they going to be inside? It seems to me that if they're inside all the time, I'm just looking at the vomit jokes now. If they're going to be inside, they have less risks. But if you have them outside, then they could be infected by birds flying overhead. I don't know. An expert would probably say to hold off. But if they're indoors, I don't see why that's an issue. I want to get an H5N1 expert. We'll see. We'll ask them that. Michael Warby has a hypothesis about the origin of 1918. How come this is never mentioned? I did an interview with Jeff Taubenberger, and he was not impressed with that hypothesis because I asked him, where did 1918 come from? He said, well, there are multiple hypotheses, but none of them are really solid. So Warby's is interesting. I've read the paper. We could have Warby on to talk about that. But the only mention it got, I think we did Warby's paper a long time ago on TWiV, so we did mention that, yeah. Have there been studies on how the immune system differs in people whose tonsils were removed versus those are intact? You know, that's a good question. The only it's two lymph nodes out of all of the ones in you. Seems to me wouldn't make a difference. But let's see if there have been any studies on that. Removing tonsils does not affect your ability to fight infections. That's from one of these, medical websites that, who knows what they're using for the source of that. So I would guess it doesn't because it's only 2 out of hundreds of lymph nodes that you have. But we should look it up more carefully. I agree. It's a good question. Thank you, Marty, for your contribution towards science communication. Yeah. Caro, hello. I remember Caro from last week. As a nephrologist, I have given repeated doses of Remdesivir to my renal patients since they are not able to receive Paxlovid. Okay, Polio Pete. Thank you, Carol. Polio Pete, if you're still here, and why wouldn't you be? There you go. You can get repeated doses of Remdesivir from a nephrologist. What more could we ask for to have experts here that answer our questions? Thank you so much. And I can't believe it's 52F there. Oh, boy. Okay, Marco has a question that maybe some of you can answer. I went to CVS to ask about Paxlovid prescription guidelines, was directed to the website. Anyone on Medicare or Medicaid can't get prescribed Paxlovid through CVS. Is there another prescription source? So if anyone's here who knows, please answer Markle. So we have 347 people tonight. So that looks like that's our new normal. We went down from 400 previous weeks. Now we're in the 350s. And we have 244 likes. So please hit the like button. Appreciate it. That gets us a little more traffic. I will do these until there's nobody here. Right? If there's one person here, I'll do this. We'll have a conversation. But if there's 0, what am I going to do? I can't talk to myself. Are there any antivirals that target the polymerase of influenza? Yeah, there's one called Bioaerosol that targets actually a component of the influenza virus polymerase, which is called an endonuclease. It is needed for priming of nucleic acid synthesis by the polymerase. The endonuclease cuts host mRNAs, and so the inhibitor Bioaerosol inhibits that. And that is the latest. That's the latest inhibitor. You'll be doing this a long time because you're going to be listening. I'll do it as long as I can, no problem. I like doing this very much. I like answering questions. I like seeing this community here. I think it can grow, and that's why I think shorts are part of it. We always have new people coming. So I think it has a good future. So here's an answer to Lise. Lise answers, Marco, it was my understanding if on Medicare or Medicaid, your physician can still order and CVS or Walgreens will fill it. Right. Would a Sarbaco virus still be a Sarbaco virus if it didn't cause a severe acute respiratory syndrome? Yes, it just has to fall within that genomic characterization, right? Because if you remember the graph, let's see if we can find it. Oh, now I have to find the paper again on my browser history. Let's see. Here we go. I found it very quickly. Nothing like a browser history. Let's bring this back up. Turn on our screen share. So, SARS beta corona. These are right here. SARS beta coronaviruses, and then we have this whole. These two branches coming off here are the Sarbacos, which are collectively put together based on amino acid or nucleotide homology, and then the Merbacos are off, and they're Merbacos because MERS. So I think that it doesn't have anything to do with the disease; it just has to do with the genomic classification. Right, what did I do here? I turned that off. Okay. All right. Any reason to get a booster polio vaccine? No, I was vaccinated in 1977-ish in elementary school. No, there's no reason. This polio vaccine works really well. You don't need a booster. But, the official CDC policy is some people should get boosters. I don't quite agree with that. I don't think there's any data that suggests that the immunity is not lifelong or long-lived. Raphael got his stickers last week. You want to see a sticker? Let's see. Here's a sticker. If I want one, just send me an email, vincent@microbe.tv. I'm happy to send you them. Why am I happy to send you them? Because it's advertisement for us, right? You put it on the back of your laptop like this, right? And, when I did the documentary, I used this laptop, but you can bet they're going to Photoshop it out. So there's no branding, right? Oh, my gosh. All right. Since SARS-2 is not going anywhere, will we ever know how long memory could last? Yes, I don't think we'll ever know. Daniela Weisskopf said that because people just keep getting infected every year. So they're getting a boost to their memory, right? So there's no way you will know how long it lasts. What we do know is after SARS-1, right? SARS-1 was around for 8,000 infection, then, it's gone. It's gone. And so you could follow people, and they have found memory T cells in those people 17 years later. So why couldn't SARS-CoV-2 memory last 17 years? It could. It's a similar virus. However, you don't need to if you're going to get infected and boosted every year, which seems to be what's happening. Even if I guess if you get a booster vaccine for 3 months, you're probably going to really severely reduce infection because your antibody levels are high, but just for 3 months or so. And then at other times of the year, you might get infected. That's the thing. We don't know what's the yearly profile of SARS-CoV-2. Right now, it's infecting all year round, right? Doesn't seem to be a winter virus. Is that going to change? Good question. But anyway, no, we'll never know. We'll absolutely never know. Sorry, I think you missed my question. No, I got your question. We don't know if it's going to be helpful. We don't know. No, I got your question. You asked, would it confer longer or broader protection? We don't know. Just because it's different doesn't mean it's going to engender longer or broader protection. We need to do the studies to find out, right? I know it's a protein-based vaccine and the mRNAs are different, but whether it's going to do something different, we have no ideas. Sorry. Yeah. Is it unreasonable to expect that a vaccine with many more T-cell epitopes and a good adjuvant could reduce severity down to the level of other human coronas? Well, I'm not sure that that's what would do it. I mean, you're saying would a better T-cell response do it? I don't know if it's all T-cells. As we talked about earlier, FC portions of antibody-mediated functions could do something as well. Not neutralizing antibodies, but antibodies that engage other cell types like NK cells. That could do it. And how do you boost that? We don't know. I think we should try. T-cell epitopes, but it's not clear to me that that's going to be the only solution. It may be that the people who get the most severe disease are mainly over 65, right? And I think part of the problem is they're infected. They have been infected for the first time later in their life. I think as the population changes, and so everyone at some point in time will have been infected at a young age, I think that's when disease really becomes modulated and becomes more like a common cold coronavirus. Because you get infected at a young age, you make a good memory response, and that's protective. But I'm speculating. I don't know. All right, another story. My father with kidney disease was treated with Remdesivir, so we now are okay with multiple Remdesivirs. You should write a book about your career and education and teaching. I have had an idea for a while to write a book. Yeah, and I have a structure in mind as well. I have a title. I don't know. I'm so engaged with everything else, right? I'm not sure where I'd get the time to do that. And the other thing is, I just cleaned out my office. I threw everything out. I didn't save a damn thing. I had files and books and everything that basically document my whole career. I threw it all out, figuring I would remember the important parts, which I probably can still do. But that's not an excuse. I do want to write one, yes, but I'm not sure when I would do it, when I'd have the time. Let's leave it at that. But I have the structure in my mind for sure. Thank you, without name or place, for your contribution. What's your take on the future of vaccinology? Will vaccines continue along the trajectory of adaptive immune responses, stepwise closer to high-affinity antibody paratopes, proliferation of plasma cells, and memory B T cells? Well, I think so. The problem with vaccinology, I think, is we focus on neutralizing antibodies because they're really easy to measure. And, the importance of T cells has been brought out quite clearly during this pandemic, but, experiments to determine their role in protection really are very hard to do, in humans anyway. So I think we have to find out a way to do that. I guess we can do it empirically. We make vaccines that stimulate good T-cell responses, and then we see if they do any better at protecting against disease. But I also think that we're understanding B-cell diversity, how B-cells react to multiple boosters and to different adjuvants and so forth. how different an antigen would be to get new memory B cells produced as opposed to expanding the previous ones. I think all of that science was going to go into future vaccinology. But I do think antigens are going to change. They're going to improve. The production methods will improve. We can engineer antibodies if you want to do passive transfer. So I think the main aspect is to get away from neutralizing antibodies as the be-all, end-all. Find some other metrics to measure when you're doing a clinical trial. Will exogenous somatic hypermutation chambers be a thing? I don't even know what that is. I don't know what an exogenous chamber is. You have to explain it. For masks, I think you need to feature on TWiV an article from Annual Review of Fluid Dynamics on the fluid dynamics of bioaerosols. Okay, well, we need to get an expert, obviously, on this. So why don't send me a link to the article, Nicola, vincent@microbe.tv. I'll start looking into it. What date in April? April 15th. April 15th. You would like to see me on Lex Fridman. I was. Les, can you please paste the link to the Fridman podcast that I appeared on? Yes, Patricia recognizes that I have been on. That was a very good episode, 3 hours plus, and it's gotten over 600,000 listens. So we've spread some word, right? I think it's good. John writes, is the increasing resolution from visible light to electron to X-ray due to the wavelength at light? Yes, the wavelength of light is quite large. So the particle has to be bigger than the wavelength. And so, well, actually, intuitively, I think that should be, but I could be wrong. But to get higher resolution of small particles like viruses, you need X-rays and you need electrons. So you need electron microscope, which gives you only so much resolution, and then X-rays for sure. I think the field of vaccinology would offer a better alternative in preventing viral infection. Well, certainly vaccines can always be improved, but they're pretty darn good right now, many of them. And VAERS is the Vaccine Adverse Events Reporting Database, where, let's say you go get a shot, and then on the way home you have an accident. So someone could write in, I had an accident, and it was caused by my flu vaccine. And so you can report stuff like that. So VAERS is full of nonsense like that, and they go through one by one and throw out the nonsense and see what's left, and there's not much left in the end. So I wouldn't worry about VAERS at all. I think it's a alert system. If something's going on, it'll percolate through VAERS, but most of the time, it's a lot of nonsense there. Yeah, so you can look at longevity of T-cell memory in other animals, but who knows what's the relationship to humans, right? Yeah, you could say a mouse which lives 2 years, the T-cells last 2 years. So what? Don't know what that means to people, and I don't think anyone can say that. What time is the TWiV on 4:15, 6 p.m.? You know, the place that I booked, the Helen Mills Theater, has a bar. I could have booked the bar, but it's very expensive. to book the bar. So here's how it works. We would have to pay. You would pay for the alcohol. And if you want beer, you'd pay $15 per person per beer. So 130 people times $15 for beer. And everyone can have beer, whatever they want. And then you could have wine too, which is probably $19 per person. So I didn't think that we wanted to do that. Just people go in. The theater is really cozy. We'll have a nice show, an hour and a half, and then we can go back to the incubator. And, presumably not everyone's going to go to the incubator because I couldn't fit 130 people in it. But those of you who are curious can come. I pulled up your 2014 lecture on H5N1. Yeah, that's pretty old. I think a lot of it's correct, but obviously the outbreak information is not up to date. But I think the fundamental science is still right. Yeah, that's right. I used to do H5N1 in my course because it was we go through cycles of being afraid of it when there are a bunch of human infections, and there was the story where they adapted H5N1 to aerosol transmission in ferrets, and that got up a lot of interest, so I would do a lecture, and then I stopped doing it. Siddhartha, thank you for your contribution to science communication. Hmm. 250,000 miles. Yeah. I spent so much money in the last few years fixing it. I tell you, though, what a good engineered car, a Beamer 328xi, 6-speed. It was in really good shape. To give me $1,000 for it, it's just a crime. Just a crime. Oh, my gosh. Thanks, Peak. Thank you for your assistance tonight. Go do your thing. Consider getting an e-bike for a 3-mile commute. I think it's 4 miles to the train station, which would take me 80 minutes, right? It's a long time to walk. I don't mind in the warm weather, but in crappy weather, and we don't really have sidewalks everywhere, so I'd be on the street a lot, and people are going to kill me, for sure. They're going to run me over. So if you guys want me to do this in the future, I shouldn't do that. Not all masks are equal. You're absolutely right. And people don't wear them correctly often. Yeah. So I agree with all. The masks are tough. You go into the train, you sit down, and then you pull your mask out and put it on. Or shouldn't you put it on before you get in? So there are all kinds of issues. We need mask education, I suppose. Yeah, Raphael says most people never use it the right way. And I think that probably in these trials adds to the uncertainty for sure. You mentioned you might doing after hours. Well, I can't use anyone's money. Music, sorry. I can't use your money to do what we're doing here. I can't use music without licensing it, but I will check it out. Roy Buchanan, after hours. Let's get a piece of paper here. I have this piece of paper from last week. You see what it says? Life with V and D. That's our title for Dixon and me, whatever we do. Roy Buchanan, called After Hours. So the idea is one of these episodes a week, I'll stay beyond 10 p.m. and just keep taking questions. And I have to warn the mods, but I won't warn you folks, so you could be pleasantly surprised. Okay. Now, listen to who's here. Mark Pelletier. Oh, my gosh, a blast from the past. How are you? Call me Vincent. Mark and I did podcasts years ago. He ran a podcast called Futures in Biotech. It was initially on the TWiV network, This Week in Tech, Leo Laporte. And Mark brought me onto that network, did a few pods with him, so Leo got to know who I was. And Mark has a day job, right? You're still in that company making aquaporin inhibitors, something like that. Anyway, you should come on to one of our pods sometime, Mark. We'd love to have you back. Mark was on a bunch of them. I think we did one which kind of a mashup of TWiV and FIB, right? Futures in Biotech. What was it called? Something TWiV, something FIB. Let me look it up. TWiV, FIB. Oh, TWiV infects FIB. TWiV 125. TWiV and Futures in Biotech joined together in a science mashup to talk about a virophage at the origin of DNA transposons and unintended spread of a recombinant retrovirus. Oh, boy. Our video was so crappy, Mark, because it was 2011. It's so much better now. Anyway, good to see you. Vincent at microbe.tv. We have a studio in New York. Mark, come by. Be fun. You look still young. Look at that. I wonder what happened to you. And you found us. How did you find us, Mark? It's cool. I had 3 Pfizers and a Moderna. I don't know how old you are. If you're, less than 65, I think you're fine. I had 3 Modernas. No, did I? I think I had 1 Moderna and 2 Pfizers. I don't remember. 3 mRNA vaccines. I'm not getting any more. If I get infected, I will take Paxlovid because I took it before, and in one day my symptoms were gone. So I don't have any comorbidities. I'm a healthy guy. And so if you have comorbidities, sure, you get boosted. But Aspie, if you're healthy and youngish or middle age, I don't think you need anything more. Right now, if it changes substantially, but I don't see that happening. You need to have a giant Zoom. Oh, you mean you want us to stream it, huh? Yeah. I don't particularly want to because there's more complications and frankly, it's more money to pay for them to do the streaming. Why don't you have the PAX money donated to Microbe? No, I don't want any money changing hands. I just don't want people to think I'm being paid off to say things about Paxlovid. I mean, I happen to agree with everything I'm going to say. That here's how it works and here's who it's for and here's what it does. I don't think there's a problem with saying that and in saying that there's no Paxlovid rebound. There's COVID rebound. I have no problem with saying that. And if it educates doctors that are not giving Paxlovid, that's good. It's not a bad thing. And if people don't like me for that, I'm sorry, but I think the word needs to get out. And using Daniel and me is a good way because we're kind of a recognized quantity, right? Is it possible to determine a mortality rate for a given virus? Oh, this is such a good question, Karen. And it is really hard. Let me show you a slide, okay? Let me show you a slide because you're right. It depends on many factors. What is the mortality rate is the number of deaths divided by the number of confirmed cases, right? Diagnosed cases. Let me show you an example of this, which underscores exactly what you're saying. Now, what lecture this would probably be in mechanisms of pathogenesis, right? So we have assembly, we have infection basics. Would it be there? All right, let's look in infection basics. That might have it. If not, we will look in mechanisms of pathogenesis. But I think it's in infection basics. I'm just opening up the file here. Is this it? No, the intrinsic and innate. No, I don't want intrinsic and innate. That's the wrong one. Sorry. Oh, here it is. This would be worth the wait. Hang on. Infection basics. So what do we look? Oh, here it is. Here it is. Very good. So everything is in my lectures. Take the course. And now we have to turn on the screen share. And here we go. Oh, good. It's not bad. Let me make it a little larger. All kinds of stuff here. Good. Now, this is my slide entitled SARS-CoV-2 1 CFR case fatality ratio does not fit all. First of all, you look at, it is early in the outbreak. 44,000 confirmed cases in China. 1,000 deaths. Overall CFR 2.3%. But look at if you break it down to age. How low it is at the young age, right? And then it's 15% over 80. So age matters. Sex also matters. Males about twice as high as in females. What else matters? How about where you are? January in Wuhan, 20% CFR. In Hubei, outside of Wuhan, much lower. Why? They didn't have as many cases. The hospitals weren't overwhelmed. Eventually, look at the rate went down in Wuhan. They got more bed space. They figured out how to treat the disease. So, yeah, it's not just the virus, folks. It is not just the virus. It is many other factors. The healthcare system, your age, comorbidities. So, really, you cannot calculate the virulence of a virus in people. Sorry, you're not doing it. You can barely do it in animals unless everything is exactly the same. And so you can't, CFR, no, the infection mortality rate may be a little more, which is a statistically produced figure, but still subject to the same criticism. Anyway, very good question, Karen. All right. Thank you very much. I'm glad you will do a PS about Paxlovid. Yeah, this is aimed at healthcare providers to make them understand that, and, Pfizer's selling plenty of Paxlovid. They don't need to sell more. They want to save lives. I believe that. And that's what Daniel and I want to do. And Leibowitz says the COVID vaccine is an IQ test. In what fashion? That if you take it, you have a high IQ? There's actually some smart, in terms of IQ, people who don't want to take it. So I'm not sure that that's all there is to it. But it's for a comedian, yeah, that's funny. Do you see a role for AI in developing new vaccines more rapidly? So here's where Amy would go off on a tangent. Okay. AI requires you to put some data in. Okay. So until we have the data on, what you need in a vaccine, I don't see AI playing a role in it. No. Okay, another question about bird feeders. I think we should ask, we should ask an expert about bird feeders. But, the birds are going to, the idea about a bird feeder is that they're congregating. But, I don't see any data that says that a bird feeder is a problem. But maybe there are such data. And I'd like to see them, but I don't know of them right now. A serotype is vaguely defined as a serologically distinguishable strain of a microorganism. That's pretty vague. It is serologically distinguishable, but how do you do the serological distinguishing? And as I said, it's originally done by prevention of disease in an animal. Oh, interesting. G. Ferraro got a YouTube survey after the Scott Hensley video. That's interesting. Some of you have also reported to me getting a survey after watching a video. So hopefully you can say good things. And I don't want to tell you what to say, but if you like it, tell them it's good. Amy's not happy that I'm answering about serotype. But I did give the right answer before, Amy. I said it was all about disease in animals, as you have taught me. I did the right thing. Alessandro Sette was elected to the American Academy of Microbiology. It's great. Well-deserved, for sure. Spamlet has suggested guests for advice on countering misinformation and cultish beliefs, Asha Rangappa and Steve Hassan. Great. Can you write those down to the yellow sticky notes, folks? I know we're thin on mods right now. We have less and Vanity and Steph, I guess. I guess that's fine. PDK left. I don't know, is Frank still here? Now we're talking about vomiting in airplanes and cruise ship. Vanity says, I tell my crew members I'll handle the medical emergencies that don't involve vomit. Yeah. And don't forget about closing the lid when you flush the toilet. It's good. Yeah, I do talk about the vomiting incident. There was an Air New Zealand flight where vomiting seemed to contribute to spread. I talk about that in my course. And Patricia tells us, wash your hands well after using a public lavatory. Yes, the toilets have no lids, so fecally transmitted pathogens are aerosolized and landing on surfaces you touch. Yes, many public toilets do not have lids, unfortunately. And so, the best ones are auto flushers, where when you walk away, they flush. Those are great, but not every place has them. Airports may have them. They're in Newark, for example. Thank you, John, for your contribution. My roommate says he's hooked after 2 weeks in a row of office hours. Well, thank you. That's very nice. I appreciate it. I enjoy doing it. I think when you enjoy something, then other people enjoy it as well. So if I didn't want to do this and I was morose and so forth, you wouldn't want to stay, right? But I like my subject, and I think it's infectious. And many people seem to like the short that I made with H5N1, so that's great. I just did it on a whim last Sunday, right after TWiV had gone up. I thought, when he said that, I thought that was a quotable piece, and I was thinking of making something short out of it. What's the other one? TikTok. Except I can't figure out how to post to TikTok. Yeah. Need to have a young person do it for me. In TWiV, Taubenberger said H1N1 spilled from humans to pigs. Yeah. And then, so in parallel, the virus has stayed in humans and pigs. And it didn't change much in pigs. And in 2009, parts of that pig H1N1 went to the genetic composition of the 2009 pandemic virus. I think it was the hemagglutinin. And the idea was that the pig HA had stayed so similar to 1918 that now in 2009, very few people had immunity to it, so it could cause a pandemic, right? And so it's drifted very, very little in pigs, and it's drifted much more in humans. And that's because, well, it may have something to do with the short lifespan of pigs and so forth. Not much immune pressure and that sort of thing. But the fact is that it has not drifted much in pigs, and that's why the 1918 HA worked in 2009 for a pandemic strain. Yeah. Memory T cells have also been found in survivors of 1918. That's absolutely right. And Hensley mentioned that, right? Is it Hensley or Henley? Now I'm Hensley. That's right. It's got an S. He said, yes, that someone pulled out memory cells. I thought they were B cells. T cells also? Well, Hensley mentioned B cells because then they made antibodies from the B cells and they were, directed against the 1918 virus. But it may also be T cells as well. Carmen, thank you for your support of science education. I really appreciate it. We need your help to do this. The incubator is an expense, and I really want this to develop. So all your help is really appreciated and needed. I keep not getting COVID. Vaxxed with 2 different types of vaccine, but I keep getting one bad cold after another. What's up? I don't know what's up. It is the respiratory virus season, and many of us who were how should I say, sheltered, locked down for a period of time, didn't get exposed to these viruses in previous years, and so we're getting them now in big numbers. And so you may just be the victim of that. There are many viruses that can cause respiratory illness besides the common cold coronas. We have the rhinoviruses, and we have para-influenza viruses, metapneumoviruses, adenoviruses, many, many more. So you could get a lot of different ones, and particularly if you were interacting with a lot of people, if you have a job where you're commuting and you're in a workplace with a lot of people, that could explain it all. Yep. A month ago, I asked why virus wouldn't phagocytose for entry similar to the mRNA vaccines. I see that the virus is designed to enter only cells where they're capable. That's very good. Yes, viruses can only get in cells that have a receptor for the virus, so they typically don't get into all cells, where the mRNA, the lipid-encapsulated mRNA vaccines can get into a lot of different cells. Yep. Good. I'm glad you watched it and figured it out. By severe disease. Well, so this is a problem, right? Severe disease. How do you measure severe disease? The studies that say severe disease often use hospitalization as a metric, and that's not often correct. What might be better is to say someone who needs a certain amount of oxygen, right? Or maybe ICU admission. So I would like a better definition. And all I'm doing for you is what I said earlier, is repeating what the paper did. They were looking at hospitalization, but I don't think it's a good metric for severe disease. Some people think it is, but Paul Offit doesn't as well. How did Scott Hensley remove only the T cells from his mice? I don't know how he did it. You could use an antibody to deplete T cells, or you can use genetically altered mice that don't produce T cells. But I forgot what he did in his paper. That was the science paper you're talking about? Let me see if I can quickly find it. Amy, do you remember how Hensley removed the T cells? Or if he removed them or genetically ablated them? Let's take a look. TWiV papers. That would be last week. That would be 983. That would be the science paper. They're pretty organized here, so I know where to look. It's good. All right, T cells. Let's see where the T cell depletion is. T cell. Okay. We depleted CD4 and CD8 T cells. So they depleted. They didn't use genetically ablated. So let's see how they depleted them. Oh, I don't have the methods section here. Yeah, I don't have the methods section. So I think that they can deplete them with an antibody. That's the best I can do there. Thank you, Kathy, for your contribution to science communication. Appreciate it. And thank you, Valwyn, for your contribution to science communication. Oh, and Valwyn has a little note here. Love to listen to your voice, your guests, your podcast, your YouTube, your personal history in virology is instructive reading. As always, you are comprehensive, objective, and articulate. Thank you. What a lovely thing to say. I appreciate it. Very nice. Oh, and so Dania is reminding me to have if you guys if you have pictures in your vaccinated T-shirts, okay. If Dania is interested in making a collage of all of you, and so Dania, where should they send them? Do you want them sent to this? Oh, I see. So Dania sent a picture of her in a vaccinated T-shirt. Here, I'll paste it in so you guys can see it. And this is the kind of picture she's thinking about. So if you want to contribute, Dania, how do you want people to send their pictures? You want to send them to me? You let me know, Dania. Put it in the chat, and then before we end, we could mention that. Will the doctors Mingarella be there on 4/15? Yes, they will both be there. Angela's coming down from Montreal, and of course, Chiara lives in New York, and so they will both be there. Yes, absolutely. For sure. Okay, thanks for answering. With respect to exogenous, somatic hypermutation chambers, I meant the engineering of high-affinity antibodies that outperform. I see. So this could be done by AI, right? But I think you need the data first in the AI before you could do that. But could you make an ex vivo system that would outperform in vivo? Gosh, I always like to say, why not? But, it may be hubris to think that we can outperform what nature has done. Our vaccines are okay, but they're not as good as an infection. It's just that, we don't want the disease for an infection. So Vanity says either send pics to me or as a comment to the Instagram link. So I put the Instagram link in the chat. If you want to send a picture there, you can do that. Or you can send it to Vincent@microbe.tv. So Vanity will make a big collage. And then what are you going to do with the collage, Vanity? What's the okay, so the point is you can take a picture of yourself with a vaccinated T-shirt, and then Vanity's going to make a collage. I don't know what she's going to do with it. She's going to put it in the chat and tell us right now. Thank you, without name or place. That's very interesting. I'm getting the bivalent in a month. Will have been a 6-month interval, which I read is optimal. Would it really make a difference if I got it now? Been 5 months. I think you can get it now if you'd like. The point is you're going to get a 3 or 4 month boost in antibody levels, which is going to inhibit infection better than when you have low antibody levels. Not totally, but it will severely reduce infection. So you're not going to get severely ill. You may get a mild cold, but that's going to go away after 3 to 4 months. So if you want that protection now, you should get it now. Should I be drunk before I watch this Lex Fridman thing? My interview with Lex Fridman? No, it was actually quite good. You don't have to be drunk. I talked good virology there on it. Yeah. Vanity said the plan is to make a mosaic-style picture of the collective photos. Okay. I was thinking of watching it after this. I have no clue who Lex Fridman is. He's a professor at MIT. He's into AI, and he has a very popular podcast. He interviews a lot of very well-known people. He interviewed me, and it was a good discussion. If you want to stay up late, Questella, go ahead. Yeah, you won't regret watching it. I had a really good time, and he had a good interview with me. Yeah. If facile people annoy you as much as they annoy me, effusive train, but he was okay with me, okay? He wasn't so bad with me. And I'm not facile, so you don't have to drink on my part, right? Oh, my gosh. Oh, we have 5 minutes left. Okay, so we have to... life with D and V is less dangerous. I know, but we want to attract people. Right, so we should do that. I want to thank Alina for your contribution to science communication. Really appreciate it. And what virus is the perfect virus? Oh, poliovirus is perfect. It's 1% away from being imperfect because it causes paralysis in 1% of people roughly. But most of the time, it hardly does anything. It's a well-adapted pathogen. It spreads efficiently from person to person, maybe some mild disease, but paralysis is an accident. So I think that's close to perfection. And look at Norovirus. It makes you vomit. It makes you throw up. What an unsophisticated virus. That is not needed for transmission. Polio shows. All you have to do is poop and you transmit really well. You don't have to have diarrhea and vomiting. So I like to compare that the noro is an unsophisticated, a highly unsophisticated version of polio. Polio is almost the perfect pathogen. And no, Elena, no way I'm keeping chickens in my house. Okay, well, I didn't mean your house, but, a coop. I guess a coop is still open to the outside. So hold off. That would be good. I suggested you call your program A Cappella Pomelo. Interesting. That most people wouldn't get it, though. Doreen, thank you for your contribution. Really appreciate it to science communication. And, gee, I'd love to hit all these other questions, but we have to stop now. I don't want to keep the mods any later. And but is this... this is our first month, February, so we need to do an after hours later in this month. You can probably guess when it's going to be. James, thank you for your contribution to science communication. Really appreciate it. And Peter S., thank you for your contribution to science communication. Thank you for your classes, discussions, Q&A interviews, and leadership in science education. Yes. You're welcome. I love doing it all. It's not a chore for me, and thank you for your appreciation. Okay, so if you want to enter this, your picture in the mosaic that Denis is going to make, this is the link. That's Denis' Instagram, and so you could send a picture there, or you could email it to me if you want to make a mosaic-style picture of the collective photos. Okay. Okay, so Vanity's taking the MCAT on Monday. Good luck. And if you get virus questions, you should be good. Any other contributions? Ian, thank you so much for your contribution. Really appreciate it. And I want to thank everybody for coming tonight. I'm very pleased that we had 350 people, 345 likes. Look at this, 345. Thank you all so much. Thank you, Les and Steph and Dania and Frank and PKD, for moderating for us tonight. We really appreciate it. We will be back next Wednesday, the 22nd of February, not office hours. That'll be Q&A with A and V. Amy Rosenfeld will be back with us, and that should be a lot of fun. 8 p.m. Eastern. That is February 22nd. Let's get the date right because I always mess them up. Yep, it's Ash Wednesday. Thank you all for bringing your wonderful questions and camaraderie to this live stream. I am Vincent Racaniello, and I will see you next time. In the meantime, please be safe. Good night.